the postdoc at Harvard Medical School and Brigham Women's Hospital, and then came down here when RI was formed in 2007. I think the building haven't got it yet at that time. And her special area is really about, because of unique training in both in the cancer and neuroscience, she actually is in the intersection between cancer and neuroscience, particularly how the cancer cell behave in the brain environment. And in this particular talk is a very quite interesting area. The micro different environment of the tumor cell in the brain environment interacting of unique neuron cells, stroma cell, astrocyte glia to the brain tumor and the brain tumor side. As you know, the brain tumor is one of the toughest cancer we have. So far, the cure is quite minimum, and it's quite devastating diseases. And this type of new approach is a really new paradigm to look at the intersection, cross-section between the neuron cell and the tumor cell and how they behave, and hopefully we can understand the mechanism better, identify the crosstalk, and possible some better biomarker and drug target. Without overdue, let me welcome Dr. Hong for her talk.
Thank you, Dr. Wang, for the introduction. And I feel grateful for so many years' support from Dr. Wang, from this group, from the Institute. And I do enjoy doing research in this multidisciplinary team and in Methodist. For these years, I have been involved in many interesting projects, but today I'm going to focusing on presenting my primary research interest in brain metastasis from triple-negative breast cancer. So I looked back my slides 8 years ago for the last promotion. The occurrence of brain metastasis is 10 to 25% of patients with cancer. So which means nowadays the incidence increased significantly. The new number showing is 40% of patients with cancer develop brain metastasis. So almost all type of cancer can develop a brain metastasis, but three major common primary cancers is from lung cancer, breast cancer, and melanoma. When patients were diagnosed with brain metastasis indicating they have a poor survival outcome, usually 2-year survival still a single digit. And clinically, it has distinct challenges because currently there are two major categories of drug treatment tailored for unique therapeutic targets in brain. One is the anti-neoplastic agents that can overcome blood-brain barrier or blood-tumor barrier. The other is targeted therapies that interrupt cancer cell microenvironmental interaction in brain. However, unfortunately, none of these has a clinical breakthrough in managing brain metastasis. It's because I think brain microenvironment with unique cell type, special anatomical structure, metabolism constraint, immune environment, imposing a distinct and profound selective pressure on tumor cells that in turn shapes the metastatic process and the therapeutic response. So as we know, for primary breast cancer, in the past 2 decades, the systemic treatment has made great progress. But however, currently oncologists confront a very particular difficult problem. Among the metastatic breast cancer patients, 50% of HER2 positive or 25 to 40% of triple negative cancers, most of them are killed by brain metastasis. The HER2 positive brain mass and the triple negative brain mass are thought to have a different mechanism. So in HER2 brain mass, tumor cells seems like to find brain as a sanctuary site because there is very limited drug accessibility to the brain. So in the HER-A clinical trial, when patients receiving the trastuzumab HER2 targeted antibody, brain made up the larger proportion of initial relapse compared with the controls. When measuring the concentration of trastuzumab in the patient CSF, it has 220-fold less in serum than in serum of the patients. And nowadays have some new small molecules like naratinib or afatinib are being developed for targeting HER2. They assume to be able to cross the BBB better, but may still be not enough because clinically the discrepancy of treatment response has been observed with extracranial disease control versus intracranial disease still progression. In triple-negative breast cancer, younger or premenopausal patients usually being reported to spread to the brain rapidly at an earlier stage or even before the primary diagnosis of cancer. So I think this indicates triple-negative breast cancer has an innate ability in adapting to the brain. So there are extensive or convincing evidence published to show triple-negative breast cancer has more cancer stem cell traits. And cancer stem cell is the driver for tumor metastasis. Nowadays, a lot of publications calling metastatic initiating cells or metastatic stem cells. And cancer stem cells or stem cells generally possess enhanced plasticity or adaptive flexibility in their cellular environment. So all these facts led us to hypothesize brain metastasis from triple-negative breast cancer may arise from a brain tropism cancer stem cell population that has enhanced plasticity to adapt the brain niche. So what is cellular plasticity? So it's in general when cells adopt different identity along a phenotypic spectrum is called as cellular plasticity. And during cancer progression, tumor cells undergo both molecularly and phenotypically change that are collectively referred as plasticity. So the most classic example is epithelial mesenchymal transition or its reverse process called MET. But emerging data have shown that cancer cell plasticity is not limited to EMT or MET. So in the following slides, I'm going to show you the research data or the evidence about existence of a cancer stem cell population in triple-negative brain mass, and also going to show you these triple-negative brain mass cancer stem cells. They have expressed brain-specific genes or acquired brain-specific properties that mediating their functional interaction with astrocytes and neurons to orchestrate their metastatic growth. And at last, I'm going to show some ongoing study about the plasticity of astrocytes in brain metastasis. So first, to show you the evidence about brain tropism cancer stem cell population in brain mass. So several years ago, we obtained fresh brain metastatic tissue from neurosurgery from triple-negative breast cancer patients. And we put them into the neurostem cell culture condition and mammosphere culture condition to let them form primary tumor spheres. And they were both successfully formed, tightly formed, condensed, round tumor sphere. So as we know, a sphere is considered to represent all progeny from a single stem cell, and the tumor sphere formation actually reflects the presence of a stem cell population. And we further propagate the primary tumor sphere by continual culturing into the secondary tumor sphere, because secondary tumor sphere formation is a hallmark of a self-renewal property of cancer stem cell population. And we found out of 22,000 the primary tumor sphere-derived cells, there were 27 or 11 spheres successfully propagated. And we used a limiting dilution analysis to estimate the stem cell frequency and found 1 out of 127 or 200 of these 2 patient-derived cells are the stem cells. So we further performed immunostaining on these tumor spheres and found they have high expression of CD44, CD133, and nesting, which are stem cell markers for breast cancer and for brain tumor. And also they express CK5. And when we put them on the high concentration of serum to induce differentiation, they lose the expression of CD44, CD133, and nesting, but acquire the expression of CD24 and increase the expression of CK5. So this data indicate there is a cancer stem cell population exists in the patient-derived tumor tissue. So the gold standard for identification of cancer stem cell is the serial tumor formation in immunocompromised animal in vivo. So we thus performed a serial intracardiac injection into nude mice using the patient-derived tumor spheres. So we have 2 patient P0 tumor. We culture them into tumor sphere to select cancer stem cell, and then 3 mice per each were injected with the 175,000 tumor sphere-derived single cells. And the P0 tumor derivative from S1 tumor was similarly cultured and injected to another 3 mice per each to give rise to S2 tumors. So we found that all the P0 tumor successfully derived S1 and then S2 tumors in all the 12 animals. And they also form intraparenchymal multiple metastatic lesions. And the tumor cells have a cuboidal shape, and they have moderate cytoplasma to nuclear ratio and have been surrounded with significant edema, and they also co-opt with the blood vessel for tumor growth, which all consistent with the clinical presentation. So immunostaining on the mouse tumor, mouse brain cells, showing they have expression of CK7, which is a commonly pathological marker for triple-negative breast cancer in patients, and they have strong expression of CD44 and nesting, and a moderate expression of CK5 and CD133. So this data confirm the tumor spheres contains stem cell population that can differentiate into the various cells in the tumor in vivo. So next, we supplement the work by using the representative human breast cancer brain-seeking cell lines. So 2 cell lines, MD231 brain-seeking or CN34 brain-seeking, both of them are originated from triple-negative breast cancer. So they were generated by in vivo selection. So these cells were injected into the mouse left ventricle, into the heart, and when they develop different organs metastases, they were dissected and cultured primarily and re-injected into the new batch of the animals. And after 6 rounds of the selection, we got the specific clone we called brain-seeking. So both of these cell lines, they derive tumor spheres. And they have been characterized as the higher efficiency forming secondary tumor spheres and have a higher percentage of stem cell frequency. Probably it's because the long time culture enriched the cancer stem cell population. But most importantly, because these cells has been stably transduced with luciferase, so we can concurrently compare the tumor sphere-derived cells versus the brain-seeking cell lines, their brain metastatic kinetics in vivo. So when we inject the same number of the sphere cells versus the brain-seeking cells into the mouse ventricle, left ventricle, the tumor sphere-derived animal has earlier detection of brain metastasis in nearly 100% of the animals. So these data indicating the sphere contains cancer stem cells have enhanced the capacity inducing brain metastasis. So next, we use the early passage of the tumor spheres derived from the two cell lines and performed RNA sequencing analysis and compared with their counterpart brain-seeking cells. And we identified 16 upregulated genes. And using the Oncology database, we found these 16 genes have a strong association with the molecular signature of human embryonic stem cell. And one of the 16 genes is called proto-cadherin 7, abbreviated as PCDH7, stood out because he is normally exclusively expressed in human central nervous system and mouse brain. You see, these are all the central nervous system regions have high expression, but they do not normally express highly in the mammary gland or in other of the bone marrow and other tissues. And also, we characterize they have a 4 to 5-fold increase expressed in the tumor spheres, and also expressed strongly in the two patient-derived tumor spheres, but not in the HER2-positive or ER-positive breast cancer cells. And they do not express in the bone-seeking cells or the lung-seeking metastatic cells from the same MB231 and CN34. But we notice they express moderately in normal mouse astrocytes and normal human astrocytes, as well as human brain blood vessel endothelium cells. So to further explore the clinical relevance of PCDH7 gene, we did a retrospective analysis on breast cancer patients, and we found the high expression of this gene correlates with the brain relapse and a shorter metastatic free survival time in a combined cohort contains 368 breast cancer patients. And we requested 29 brain metastasis patient tissue from the Methodist tissue bank. And out of the 29, 90% of them have high expression in the brain metastatic lesion, but not in the lung mass. So this indicating PCDH7 may be a key contributor for brain metastasis. And to further determine the functional role of PCDH7, we use the animal experiments and by injecting the shRNA, knock down the PCDH7 tumor cells into the left ventricle. And we found by the end of 4 weeks, almost all the animals injected with the tumor sphere with a control shRNA group, they develop a brain metastasis. But until week 6, we see 40% of the animals with the shRNA knockdown animal develop brain mass. At the end of 8 weeks, still 40% of animals, they didn't have brain mass. The similar delay or prevention of brain mass were observed in the second animal model. And so this indicating the high expression of PCDH7 in tumor cells have the higher capacity to form brain metastasis in animals. So to further study which step that PCDH7 contribute most significantly to brain metastasis, we first did an in vitro blood-brain barrier assay, but we didn't find significant effects of knockdown of PCDH7 affect the tumor cells we called penetrate through the BBB. But using this intravital two-photon microscopy imaging, we found that the knockdown cells showed a significant defects in the tumor cell colonization in the brain. So in these experiments, we used a drill to carefully make a thinning skull window on the mouse brain to make the skull be transparent, but still maximally keep the physiology of the brain blood or the CSF circulation. So in this case, we can imaging the blood vessel as well as the tumor cells by the fluorescent dye. So in the control animals, by 2 weeks, we see 70% of the extravasated tumor cells actually they form multiple cell loci, contains more than four cell type, more than 4 cells. But in the cells we injected with the PCD7 knockdown cells, either 75% of the cells disappear, of the extravasated tumor cells disappear, or 15% of the cells keep the solitary status without further progression. So indicating PCDH7 has an exclusive role in mediating the tumor cell colonization. However, in vitro, when we put these cells in culture, we monitor their proliferation, we don't see any proliferation change between the knockdown cells and the control cells. So this promoting us to investigate the brain microenvironment elements that facilitate the PCD7 mediated brain colonization. So we found this data set in the GEO. The experiments was done by co-culture the parental MB231 cells with astrocytes. So when they been put together for 48 hours, PCD7 was one of the genes found significantly upregulated in the tumor cells. And we also noticed this gene was moderately expressed in mouse and human astrocytes. So we put these 2 cells co-culture, and we found the human astrocyte actually facilitates the retention of the tumor sphere self-renew. And the tumor spheres could be propagated beyond the passage 5 when cultured with human astrocytes, but the sphere cells variability was diminished by passage 2 when co-cultured with the control normal fibroblast. And we further use neutralizing PCD7 antibody. It not just breaks the heterotypic sphere of these astrocyte and tumor cell sphere, but also inhibited the propagation of the spheres in vitro. So in general, proto-cadherin family are thought to mediate the cell-cell adhesion. So in recent years, PCDH7 has been shown to mediate the tumor and astrocytes assemble a gap junction. But more research showing the proto-cadherin family are more important to relay the signal to their cytoplasma. So which means PCDH7 may not only mediate cell-cell adhesion. To prove this or to explore this, we transfused either the full-length PCD7 gene or only the extracellular domain PCDH7 gene into the parental breast cancer cells. And in the animal experiments, on day 3, we can see exclusive and strong astrocyte activation in the proximity to the brain metastatic cells. So when we quantify the astrocyte reaction in the animal's mouse brain, to interacting with either the full length or the only extracellular domain of the transduced tumor cells, we actually found a similar astrocyte response in these 2 groups. But at the end of the 4 week, we see obvious defects in the tumor colonization with the extracellular domain tumor cell injected cells. 90% of the full-length tumor cells form more than 5 cells cluster, but only 22% of these groups have colonization. So this indicating the cytoplasmic signal are essential for brain colonization. So PCD7 protein are majorly located at the cell membrane, but some are in nuclear and cytoplasma. So similar as some other proto-cadherin family members, PCD7 has a small serine-rich domain in its C-terminal that is homologous to the beta-catenin binding site of classical cadherin. So which means Wnt signaling pathway may be involved as a cytoplasmic signal. We do use the IP to find PCD7 can weakly bind with beta-catenin, but when we put the tumor spheres co-culture with the astrocyte, we don't find any significant nuclear translocation of beta-catenin and neither the activation of the key transcript factor. So this results indicating the canonical Wnt signaling was not functional in enhancing brain metastatic tumor cell colonization. So because PCD7 mediates the gap junction of astrocytes and tumor cells, facilitating the calcium signal being transmitted, and extensive data have indicating the persistent calcium increase stimulate tumor cell proliferation, in contrast to the transient calcium increase actually activate mitochondrial apoptotic pathway. So we perform live cell calcium imaging. So we found the tumor spheres, actually cells, they have increased the cytoplasmic calcium, about a 30% increase at the baseline, and dependent of the PCD7 expression in the tumor sphere cells. So when upon the influx of calcium from astrocyte to tumor cells, we noticed activation of an enzyme called phosphodiesterase on the cell membrane, and the downstream calmodulin dependent protein kinase 2, as well as S100A4, these downstream signalings. So this enzyme abbreviated as PLC, it characterizes the PIP2 to become IP3 and DAG. So both of these signaling activation are important to increase intracellular calcium storage upon the induction of the transient calcium influx. And they further activate the transcript factor that's stimulating the maintenance of pluripotency of stem cells and proliferating. To further prove this signaling is important, we reposition a clinical drug. It's called idelfosine. It's a specific PLC inhibitor being used as bone marrow transplant purging drugs. And we applied these drugs to the animal models. So to mimic the clinical situation of brain metastasis in patients, where brain metastasis were established before treatment, we starting the treatment at day 11. So let the tumor formed in the animal brain for 10 days. We observed micrometastasis in the mouse, and then the treatment lasts for 15 days. So during the 15 days, the treatment significantly suppressed tumor growth, especially the macrometastasis was suppressed nearly 90%. And the intracellular enzyme activation and the proliferating were significantly suppressed. And due to the limited availability of the patient tumor spheres, we only did in vitro treatment and found the dose-dependent inhibition of the secondary tumor sphere formation in the patient-derived tumor spheres. And we see a significant suppress of the phosphorelated enzyme. So these results indicating the potential of targeting the PCD7 PLC signaling inhibit the brain metastatic growth. So in addition to the PCD7 story, GABA is one of the most abundant neurotransmitter in brain. We found actually several GABA receptors like GABA-R and GABA transporter like the GAT, as well as a GABA synthesis enzyme called GABA transaminase ABAT, are significantly upregulated in the tumor spheres and as well as in the brain-seeking cells. And we exogenously applied the GABA to the in vitro culture and observed a dose-dependent increase of the tumor cell proliferation with EC50 around 0.2 micromolar. So the increase of the tumor cells proliferation may be due to two reasons. One is probably due to the GABA receptor signaling. The other is maybe due to the increased GABA metabolism. To differentiate these two possibilities, we used a compound called mucimol. It's a potent GABA mimetic, selectively bind to GABA receptor, but could not be used as metabolic source. So upon adding this compound to the culture, we didn't find any proliferation change in the cells, so indicating excluding the GABA receptor signaling mediating the tumor growth increase. To further confirm that the metabolic pathways are be important, we use another compound. It's called vigabatrin. It's an irreversible inhibitor of ABTA. It's a GABA synthesis enzyme. We do see this compound inhibit the GABA-induced tumor cell proliferation. So demonstrate the GABA is the source of energy that the triple-negative brain-seeking cells can use to fuel their proliferation. Start moving. Okay. So in some brain regions, the GABA concentration can be as high as 1 micromolar level. So with the high expression of GABA transporter, this high concentration of GABA being transported inside of the tumor cells, and upon the catalysis of ABTA, and convert GABA into succinate, and resulting in subsequent production of NADH to satisfy the energy and growth requirement of the tumor cells. So this process is called GABA shunt. It's an in vivo metabolism pathway in the brain cells. And when we measure the intracellular NADH concentration, we do see GABA, exogenous GABA, increase 1.5-fold increase of the concentration. But when we apply the vigabatrin, it suppresses the NADH. So clearly indicating the triple-negative breast cancer brain metastatic cells co-opt GABA as an oncometabolite. So glutamate is another important neurotransmitter in the brain in addition to GABA. So we also found several glutamate receptors being highly expressed in the brain metastatic cells as well as the tumor spheres. And also, you see, compare with different subtype of breast cancer, the basal-like as well as the triple-negative has the highest NMDA receptor score, especially this GLU-N2B component. And the high expression of this component correlates with the decreased metastasis-free survival in triple-negative breast cancer patients. So when inject the cells, the brain-seeking cells, into the tailwind to induce lung metastasis, or to the mammary gland for primary tumor, or the intracardiac for the lung, for the brain metastasis, it's only brain has the activation of this component, not the lung, not the primary. So indicating this component of the NMDA receptor is important to brain metastasis. And next, using the neuroscience approach, the PET clamp, the exogenous NMDA was applied through this puff application by this left pipette, and used the left pipette to record the single channel current, and detected about 4.5 PA amplitude, and equivalent to the cord conductance of 50 p-second. That's the characteristic of this component in the normal brain cells. So this data indicating this receptor has a function in the brain metastatic tumor cells, but GABA can be either autocrine through the tumor cells itself to activating this receptor, or can be paracrine secreted from other brain cells to activate this receptor. But the condition media was collected to measure the concentration of glutamate from the tumor cells, and there is no significant change, so indicating the autocrine glutamate is not important, suggesting an paracrine glutamate supply. So using a super resolution microscope, the immunostained mouse brain slides with brain metastatic lesion. So the blue is the luciferase staining, is the tumor cells. The red is the presynaptic excitatory neurons staining, and the green is the postsynaptic, the receptor signaling. So this diameter is 400 nanometer. So this indicating a normal synapse formed between the pre and post synaptic neurons, and this indicating the brain metastatic tumor cells have closity to the pre and post synaptics, and further use an electron microscope to show the protrusion of tumor cells has a proximity to pre and postsynaptic dendrite or axon with excitatory neurons. So then it's been proposed that the tumor cells form a pseudo tripartite synapse with the neurons to get the supply of glutamate, which are normally released from the presynaptic neurons, and to make their own activate the NMDA receptor to sustain their proliferation. So normally, astrocytes located at the synapse cleft area, they quickly absorb the glutamate being released from the presynapse. And now metastatic, the brain mass breast cancer cells take over this position and co-opt with the neurons to sustain their low proliferation, and further use a conditionally knockdown of this receptor, showing a dramatic decrease of the brain metastatic colonization. So in summary of the tumor astrocyte neuron orchestration in the brain metastatic cascade, we see PCT7 mediate gap junction, facilitating the calcium signaling, activating PLC and the downstream transcription factor to sustain the tumor cell proliferation, and in the meantime activate GABA shunt to have oncometabolite to fuel the GABA, to fuel the tumor cells, and also co-opt with the neurons to take over the glutamate to activate an NMDA receptor for this brain colonization. So until now, I've been showing you the evidence of the existence of cancer stem cell population, and they express brain-specific genes or acquire brain-specific properties, and these functional mediate the interaction with both of these two brain cells for their metastatic growth. And lastly, I want to show you some recent data about astrocyte plasticity in brain mass. So astrocyte activation happens in all brain disorders, but in compared to the acute brain disease condition like a stroke or TBI, the brain metastasis is a continuous progressing insult. So at this time, the astrocyte could not defend the insulting brain metastatic cells. Along the time, they were hijacked by the tumor cells and to functional benefit the tumor cells growth. So in these experiments, the top is showing the activation of astrocytes. So we see astrocytes have a two-phase activation. We call acute and a later. But this two-phase activation may not have the same function. And some existing paper or research is showing the opposite behavior of astrocytes, depending on the disease stage. And we see at day 3, for the tumor growth, the tumor already being inhibited compared with right after injection. But then later, at later stage, the tumor regrows. So probably indicating the astrocyte acts as the innate host defense system to limiting the progression of the tumor cells, but later favor it. So when we're trying to study astrocytes, especially to perform the functional study, we need to have a better culture condition. But the traditional astrocyte culture preparation from the 1980s protocol was mostly from the rodent neonatal brain. It has several limitations. First, it's very tedious. This whole process can take over a week or more. And secondly, these cells divided rapidly or continuously. But in vivo, astrocytes is pretty much quiescent cells. And third, they cannot be obtained from a postnatal adult brain. The cells will die. And fourth, these isolated cells, they must be cultured in serum-containing medium. But we know in vivo, except the brain has an injury, the astrocytes will no longer be exposed to the serum. And lastly, when perform the gene expression, these cells, astrocytes from the neonatal brain, has different gene expression profile compared with acute isolated adult astrocytes. So we use a protocol to isolate astrocytes called immunopanning. So immunopanning basically is use a succession of negative plates to get removal of the different cell types from the brain suspension, including microglia, macrophage, endothelium cells, and oligoprogenitor cells, oligodendroid progenitor cells, and use the last plates coated with specific antibodies to bind with the astrocytes. For mouse, it's called IgG5. For human, it's called hypercam5. So in this way, the whole process can be done within 1 day, and the process is very gentle, so we can isolate the astrocytes and keep them continual culture for 2 weeks. So using this protocol, we harvest 90% of the purity of the astrocyte from the adult mouse brain. And it's been published in this group that they also get the astrocyte from the human glioblastoma tissue. So next, we inject the tumor cells to the mouse, and after 3 days, on day 3, because I've been showing you the astrocyte activation has a day 3, the first phase. We isolate those astrocytes as well as the tumor cells and performed RNA-seq. And we're trying to use the CCC Explorer, which are in-house developed software is for studying cell-cell communication, to study how these astrocytes interacting with the tumor cells. So in CCC Explorer, we are able to study 4 types of interaction network. One is astrocyte to tumor as a paracrine, or astrocyte to astrocyte autocrine, or tumor to astrocyte paracrine, or tumor to tumor autocrine signalings. So this is a screenshot of the CCC Explorer when we use astrocytes data and the tumor cells data to explore the astrocyte to tumor paracrine signaling. So these red nodes are the ligand, possibly can be secreted through the astrocyte. The yellow dots are the receptors located on the membrane of the tumor cells. And those green are the intracellular signaling molecules. And these blue are the transcript factors in the nucleus. So we have output as significantly changed pathways mediating their paracrine interaction of the ligand receptors. So in specific, we found 27 ligands from astrocytes, 30 receptors on tumor cells, and 8 transcription factors within the tumor cells, and 9 significant activated tumor signaling pathways by this crosstalk. And we noted very few active paracrine or autocrine signaling from tumor to astrocytes or tumor to tumor at day 3. We call early stage brain mass. But we found many more signaling involving astrocyte to tumor paracrine and astrocyte to astrocyte autocrine. And most of the astrocyte autocrine pathways and the paracrine pathway are potential novel inducers in BM because we didn't find any existing publication. To further prove the astrocyte do being activated and they can secrete the ligand and interacting with the ligand in the tumor cells, we immunopine astrocyte, we culture them, and we did ELISA to measure the cytokines being released from the astrocytes, from the mouse astrocytes. So in this assay, one time we measured 40 mouse cytokines. So but we injected 2 types of cancer cells into the mouse. One is breast, one is lung. But we found 7 significantly changed cytokines in the astrocytes, and 6 of them are overlapped between lung cancer and brain cancer induced brain mass. So this indicating even at a very early stage, the astrocytes, functionally they can secrete cytokines. And this is probably ubiquitous to the tumor cell infiltration because lung and breast have the similar cytokine release. And further, the cytokine supposed to be interacting with the receptors located on the tumor cells. To explore the clinical relevance of these cytokines and the receptors, we found data sets from the GEO contains 204 breast cancer patients with their known site of relapse, which are focusing on brain relapse. And we found the interleukin-6 ligand, which is IL-6R, and CXCR5 receptor CCR5, and the PCD7 receptor EGFR have a significantly correlation with decreased brain metastasis-free survival. So this showing the clinical relevance, and next we're trying to validate the computational predicted results. We performed in vitro co-culture. So 2 types of assay were established. One is we co-culture them indirectly, which means we use a conditioned media to basically look for the cytokines in the conditioned media, mediate this crosstalk. The second, we use the co-culture directly because the ligand can mediate cell-cell contacts. So either metastatic brains or normal brains will undergo immunopanning to isolate astrocytes. And from this tumor, we also get tumor cells. When we collect conditioned media, we applied to the culture of the astrocytes, and we look at the concentration of interleukin-6 or CCR5, and we found significant increase of the astrocytes. And we collect this control condition media again to culture the tumor cells. We found the tumor cells intracellular signaling downstream to the interleukin-6R have significantly upregulation or the CCR5 signaling. For the direct co-culture, we found the tumor cell directly co-culture with astrocytes actually activated this EGFR and the downstream signaling. And we do see the strong expression of PCD7 in astrocytes as well as the tumor cells. So this data indicating the plasticity of astrocytes in response to brain metastatic tumor growth. So we are continuing to explore the in vivo cell-cell crosstalk of these identified astrocyte to tumor paracrine signalings using conditionally knockdown of astrocyte genes in animal model. And we also assessing the astrocyte-secreted proteins in modifying the function of BBB and the microglia macrophage in the early stage brain metastasis. The motivation is because we see at the early stage most of the tumor cells is still inside of the blood vessel, which means they have not fully extravasated from the BBB. But astrocyte-secreted proteins, they're supposed to have the diffusion diameter of more than 250 microns. So to study this is also meaningful because although we know a lot of information about the BBB in healthy brain, but little is known about the BBB in the brain disorder, especially in brain metastasis. So which is first challenging for the clinical drug development. So the third one is we further characterize the temporal evolved astrocyte to brain to tumor cells crosstalk in a cancer cell specific fashion. And to make the conclusion, TNBC breast cancer cells functionally interact with both neurons and astrocytes to alter their metastatic growth in the brain. And the cellular plasticity of both tumor cells and brain cells are detrimental to the tumor progression. I want to acknowledge the founding source, and especially Dr. Wang's group, including the previous and the current lab members. I feel be very pleasant to remember the previous members and also the collaborators in Cancer Center, in MD Anderson, in Weill Cornell and Baylor College of Medicine. And lastly, I want to thank you to be in this room. Thank you very much. Question?
No question? Any other question? So you think the plasticity through the astrocyte?
Tumor cell has plasticity for sure.
Yeah.
And astrocyte also has plasticity. And I would assume neurons and microglia, macrophage, they also have plasticity.
That's something to do with, you look at the tumor heterogeneity. They can be inter or intra.
Yes. Mm-hmm. Yes.
There will be inter-tumor heterogeneity you talk about here. I wonder whether for intra-tumor
yes.
heterogeneity.
Yes.
You deal with that.
I show you one. Oh, yeah. Actually, it's a little bit small, but intra-tumor heterogeneity is significant because for these cells, we inject through the left ventricle at the same time. But their development into multiple cellular cluster is quite different at 1 month later after injection. We still see a lot of single cells solitary in the mouse brain, but some already developed into the micrometastasis containing more than 50 cells, indicating they have intra-cell heterogeneity, right?
Yeah.
Mm.
Actually, interesting. Seems like you don't mention too much about EMT, MET in the brain mass. In a lung area, in other area, there's a lot mentioned in the area, but seems like in this area, it's not quite. It's different because
it's different because of triple negative.
Oh,
triple negative breast cancer cells, they are M type. They are not E type.
Oh, I see.
Yeah. So PCDH7 is a member of cadherin family, right? But it mediates cell-cell contact adhesion, but not 100% identical to the E-cadherin function. And also in the brain metastasis, there's a lot of expression for N-cadherin, which is also the M marker. It's not an E marker. So PCDH7 may possibly interacting with N-cadherin to form the heterotypic dimer for the cell-cell interaction. But I don't examine E-cadherin expression in the brain. Yes, I recently changed the different antibodies, and I didn't find any positive staining for the E-cadherin.
tumor behaves differently in different organs. Quite interesting on the metastatic. Yeah.
Thank you.
Thank you very much. Wonderful talk.
Oh, we stopped sharing screen.
Yeah.
I stopped sharing the
You
screen.
can stop sharing that.
Yeah, I stopped already.
Control.